Right. The great thing about organizing this HBES conference is that you get to select the keynotes, and because it's an interdisciplinary field, you can pick the best ones in that field. And I think we have one of those. So I'm very pleased to introduce to you Professor Toby Kiers. She works actually at our university, the VU University of Amsterdam, as a professor in evolutionary biology, and she also holds a university research chair there. Toby did her PhD at the University of California and a postdoc at Amherst before coming to Amsterdam, and we're obviously very happy that she works here. Like many of us, she studies cooperation and the evolution of cooperation, but her research participants are a little different from those that you and I use. She specializes in understanding mutualistic relationships between plants and fungi, and she uses Darwinian concepts like biological markets and punishment, of course, to understand what happens in life's undergrowth. She also has a specialized interest, I'm not sure if she's going to talk about it today, but in Darwinian agriculture. And that's a very interesting field because it is about new and innovating ways to do farming science. Are you going to talk about that today?
I'm not going to talk about that.
Oh, well, next time then.
Next time.
Her work has been published in all the top journals in the field, Science and Nature, etc. Please give her a warm applause from all of us.
Hello, Amsterdam. So can you hear me okay? Is the sound good? Okay, so I have two sort of goals in this keynote lecture today. And the first one is to convince you the importance of cheating in nature. And the second one is to try to have you think about dirt a little bit differently and kind of see why somebody would want to study underground. So I want to start with a series of images. I want you to tell me what these images all have in common. Besides, they're all black and white, they all involve children, but there's a key aspect about these images in particular, where they all show children being sucked underground. So there's this photographer, Arthur Hess, who took photographs in the 1960s, and what Arthur Hess did was he tried to recreate the nightmares of children. And then he'd photograph them. So he'd have them describe the nightmares, and then he'd photograph the children in their nightmares. And what he found was that there was this link, there was this commonality between children's nightmares, that they had this common theme of being pulled underground. So why this fear of the underground? Well, one of the reasons is that we probably, it's very unknown. It's very unknown and it's very scary. I mean, people say that we know more about space than we do about what's underground. And this is what's fascinating. So what we do know about underground is that we see these extraordinary networks that are populated by microbes that have evolved strategies to manipulate each other and their environment. And this, as an evolutionary biologist, this is what I'm interested in, this behavioral component of microbes. So as Mark said, I study the evolution of cooperation, or at least that's what I used to study, until I realized actually that cooperation is quite sort of boring compared to the other side of cooperation. And cooperation ends up kind of looking like a dull cousin when you start studying conflict. I mean, that's where all the tension is. That's where all the interesting biology is. Now, of course, that's because organisms are incredibly selfish, right? This is sort of the mainstay of evolutionary theory. But this selfishness can actually lead to some really interesting emergence of different strategies. So selfishness can actually drive the emergence of cooperative partnerships. So I think the example of cleaner fish and their clients is sort of one of the most elegant to illustrate this, where you have a client fish that's covered with ectoparasites, and the cleaner fish gets a meal by eating those ectoparasites, and of course the client fish is cleaned of those parasites. So they both have a selfish interest in forming this partnership. But what I'm actually interested in, and I love it, it looks like a deer caught in headlights, like, oh, we got caught. I like this picture because what I'm interested in is the organisms that actually take a bigger bite, right? So they don't just eat the ectoparasite, but instead they also get a bit of that flesh. And why not? I mean, if you can get the parasite, you can get a tiny bit more, why not defect? So there's clear advantages to this kind of behavior. There's clear advantages to cheating. And when you think about it, I mean, it really is a very simple sort of cost-benefit analysis in terms of determining strategies. There's the probability of getting caught. There's how much that is actually gained. If there's nothing gained from cheating, then it can't emerge. And then there's the question of the punishment, right? And how severe is the punishment? And is it worth the risk? And in many cases, the answer is yes. And in many cases, the best strategy is actually to defect from these cooperative partnerships that you're involved in. But of course, cheating is also a social art, and it really depends on the frequency of other cheaters in that community, right? So it's a frequency-dependent behavior. So I really like this graph. It's quite elegant, where it just shows proportion of cheats on the x-axis, and then the relative fitness of the cheat on the y-axis. And what you see is that as the frequency of cheaters increases, right, then the relative fitness of those cheats decreases. So what that tells us is this very social behavior, and it really depends on what others in the population are doing. So what we do in my lab is we actually try to create conflict, right? We're like rebel rousers. We try to find these beautiful, ancient partnerships and then actually try to drive a wedge into them and create conflict and see how the other partners respond. Right? That's a way where you can study sort of punishment and conflict, is if you create it yourself. So what we do is we try to manipulate levels of cooperations in these tractable systems. So we try to force individuals to cheat, and then we test how the other partner responds. So getting back to what's happening on the underground, this is actually a really nice place to study the evolution of cooperation and conflict, because there's this inherent tension. And I think this is, if one thing to take away from this talk, is it's sort of in these next couple slides. And that's that there's tension, right? If you imagine a plant, and it's got a root system, of course the microbes associated with that plant, they benefit from being on a healthy host, right? So you can imagine, sure, there's a direct benefit from making the host healthy if you also benefit. But we've also got a real potential tragedy of the commons here, because what you have is you've got microbes, let's say on a root system, that are potentially benefiting from the general benefits of being on a healthy host while paying none or less of the costs. So these are the sort of microbes that I'm interested in. So what that means is that we need to look at two levels of tension, right? There's the tension that we usually think about in partnerships between the host and the microbe, and both are competing, don't want to give it as much resources, but they'll give enough. But there's also a conflict among the symbionts, multiple symbionts colonizing that same host. So there's conflict between the host and the symbiont, but there's also conflict among those symbionts, because they're all trying to benefit from the healthy host without giving much in return. So what we're trying to understand is how that conflict is actually managed. So one of the things that we see repeatedly, at least in nature, is the evolution of top-down control, right? Top-down control is sort of, a really intuitive sort of answer to this question of how to manage conflict. And so some of the work that we've done looks at, for instance, nitrogen-fixing bacteria, right? These are bacteria that grow on the root systems of legumes, and they take atmospheric nitrogen and they turn it into a form that the plant can use. And in return, the plant gives those bacteria sugars and feeds them. But what we try to do, for instance, is we force those rhizobia to cheat. We don't allow them to fix nitrogen by, for instance, growing them in a nitrogen-free atmosphere, and then we see how the plant responds. Now, of course, this is an example of top-down control, and what we see is that the plants can actually sense at the level of a single nodule. They're colonized by many, many different strains of rhizobia on a single plant. But down to the single nodule, the plant can sense how much nitrogen is coming and actually suffocate those bacteria that fail to give nitrogen. But we see this in other examples in nature as well. So the legume rhizobia is a very nice example, but protists are also a great example that rely on algae for their photosynthate. And for instance, if the algae, let's say the conditions are under low light, and so the algae are not giving as much photosynthate as necessary, the protists, they digest them. They digest the algal symbionts. It's another form of punishment. For instance, squid rely on bioluminescent bacteria for anti-predation control. And they use the light formed by this bioluminescent bacteria. But again, they've evolved these mechanisms to expel single bacterium if they fail to give light. So what this tells us is that hosts have evolved very, very fine levels of discrimination among the symbionts that they interact with. But what we see is that it's not, there's sort of two commonalities that we have to think about in terms of this host, this top-down control. We see it when there's multiple competing symbionts. And again, this is the thing I really want you to take home is this idea that we've got multiple genotypes on a single host, right? And so as soon as you see that situation, that's when we have to start thinking about how punishment mechanisms can evolve. But we also see it when there's lots of environmental variation, for instance. So the environment is not so stable. So as soon as there's changes in context, the host has to be able to respond to those changes in context and be flexible in terms of working with different symbionts. So one of the solutions that we see in nature is evolving the ability to compartmentalize partners, right? This is actually a really elegant solution. If the host can separate their partners into different compartments, then they achieve two goals at once. One, they prevent them from directly competing with each other, and two, they've evolved a way where they can discriminate amongst them by having them spatially separated. So again, nodules are a great example where, of a solution of compartmentalization, where legumes have evolved the ability to physically separate a single bacterium from another bacterium and let them proliferate in very specific structures so that there's no conflict among them. So again, what this does is it gives a physical separation so you can decrease conflict among competitors, but it also allows for discrimination. So you can sense where you're getting your resources from, and then potentially, for instance, in the case of legumes, suffocate the bacteria in the nodules that aren't providing enough. But this has taken millions and millions of years of evolution, right? I mean, the example that we're giving now, 60 million years old, this symbiosis. But over that time, we've seen the way that the host can control the rhizobial fitness. They can control how much oxygen they get, how much sugars they get. One of the really sort of interesting ones that we're studying right now is how they can just get one single rhizobia strain per nodule. Because there's some legumes that don't show that ability, where a nodule actually contains multiple strains. And those are very interesting too, because there's some evidence that even if you've got two genotypes in a single nodule, that the host can discriminate within that nodule and actually target specific genotypes. So again, 60 million years of evolution to get this kind of resolution. Now in some cases, this top-down control seems to go in such an intense directional selection that what we see is that a new organism evolves. And this is what we call where you actually transcend sort of the original levels of individuality. So what we're trying to understand is you've got these partnerships, and they're on these evolutionary trajectories. So how and when do these partnerships that were once free living organisms become integrated into a single organism? So in those cases, not only do we see a compartmentalization, but we see such extreme dependence that they become a single organism. Mitochondria are a perfect example, our own mitochondria, of a free living organism that's been integrated into become a new organism. So again, the evolution of control, what we see is that it mirrors, in many cases, this evolution of increased dependence. But not all the time, and that's what actually makes it so interesting to study. So what we're trying to do now is get a really broad survey and look at these host-symbiont relationships and try to understand if we can predict what kinds of cooperative relationships emerge in different particular trajectories. So what we're trying to make right now is what's called the Global Symbiosis Database, where we're looking at host and symbiont partnerships and trying to understand their environment, their function, whether the hosts have some commonality that can drive particular trajectories of dependence, transmission mode. So again, transmission mode is incredibly important, right? You can either pick up your symbiont every generation from the environment, anew, or you can pass it down vertically, generation to generation. And that can have a really big impact on the level of dependence that you evolve with your symbiont. So we took our first stab at trying to understand there's sort of these general patterns over the tree of life by looking to see if we could make any predictions about how particular trajectories were evolving. Now what you're looking at here is a phylogeny of bacteria, and then surrounding it are the different hosts that host those, make relationships with those bacteria. You've got the red dots, and that's where you see vertical transmission, and you've got the black dots, and that's where the organisms are picking up their symbionts from the environment. And what we see is that function, so function is incredibly important. What is the symbiont doing? That seems to determine whether hosts become more or less dependent on that partner. So we see the greatest dependence on symbionts when they provide some kind of nutrient and when they're vertically transmitted. So from generation to generation, which also makes sense. There are also really interesting examples that don't follow this pattern. For instance, in marine systems are, it's like going to a different planet, right? When you start looking at marine systems, you can have hosts that are obligately dependent on a nutrient-providing symbiont, but yet they're picking them up horizontally from the environment. So we see this incredible dependence that you only see in terrestrial symbioses, yet they're still picking up the symbionts from the environment. But in general, that's what we saw. We see a greater dependence on symbionts that provide nutrients. Now, that actually makes sense, right? Because nutrients are something that are not as context dependent as, say, defense, right? A lot of symbionts provide the service of defense. But nutrients we need more consistently. So this brings up a really interesting question about, well, what does it mean to become dependent, so dependent on another partner that you actually merge into a single individual? It's hard to study these because, I mean, how do you do it? As soon as it's very difficult to sort of tease apart the difference between dependence and benefit. Right? Imagine you become so dependent on a symbiont, again, mitochondria is a great example, that you remove it. Of course there's going to be some sort of fitness cost, but does that mean you're just dependent on it, or does it bring some sort of benefit? So again, teasing apart these differences is quite hard, but this is what we see. This is one of the patterns, the overriding patterns that we see about cooperation in nature, is that you can drive this dependence, and maybe it's even hard to, in retrospect, identify what advantage that dependence brought. Yet we still see it. So does that mean that the partners are just driven into a rabbit hole, or that we don't understand the actual function of those symbionts yet? Okay, so the first half of my talk has been very focused on hosts, right? And how hosts become dependent on their symbionts. But what actually I want to do for the rest of the talk is sort of turn it around and look at the microbes themselves, right? Because really, when we think about this, this is where the most interesting dynamics emerge, is when we look at the microbes themselves. Now, we're actually in a really interesting period in biology right now. I mean, I think it's a time when paradigms about our understandings of microbes are really, really shifting. And actually, I think this is where a lot of the human behavior science can come in handy. So So the old view of microbes were really as standalone asocial organisms, right? But we're in this renaissance now. We're understanding microbes as social actors, right? Who can form sort of complex networks of very coordinated behavior. And we're really interested in trying to understand how these types of behaviors emerge. So for instance, just some examples of the kinds of things that we see in this new field of what we're calling social microbiology is that we can see microbes can hunt in packs, they can jointly, so together, scavenge for resources. They can form very complex structures where some of them actually sacrifice themselves. They can collectively conserve resources, right? I mean, as humans, we can't even do that. Yet somehow they've evolved this ability to do it. So some of the things that we're interested in studying these sort of microbial behaviors is, okay, well, how can you have effective communication? How can you coordinate groups at such a massive scale? Right? I mean, it makes the populations, the global populations look minuscule compared to the kinds of individual levels that we're talking about of coordinated behavior in microbes. And again, prudent use of resources, right? The ability to actually save resources for a different time. Now, we're also seeing this huge renaissance in studying the microbiome. And I think as evolutionary biologists, and I'm really, getting on a pulpit here, but this is really important to me. I think we have to be really careful in this age of everybody being so dazzled by the microbiome. The microbiome is incredibly, incredibly important, and we're just starting to understand. I mean, so this is a number, for instance, everybody's always talking about what is it? Humans aren't really humans because 1 in every 10 cells is bacterial, things like that. Plants, it's even more intense, right? So plants are only 0.1% plant cells. The rest are other types of cells. And there's a lot of important work that's been done on the microbiome that's showing how it actually is an adaptive immune system. So it actually has the ability to expand the genomic capacity of the host immensely. Here are just a few: enhanced host immunity, novel nutrient acquisition systems, improved host architecture, tolerance to stress. The microbiome can do it all, right? Sign here. Everybody is on board. Everybody loves the microbiome. But I think the problem, and what we need to think of as evolutionary biologists, is that we have to separate importance from harmonious. Right? So the microbiome is not there necessarily for the plant's benefit. And this is a difficult lesson to learn, especially in the human microbiome studies. But I think a lot of the work assumes this sort of harmonious relationships, but actually microbes they're the masters of manipulation, right? And they're under natural selection, just like the host is, and they're also trying to maximize their benefits. And many times, those benefits align with that of the host, right? And then that's start of a beautiful cooperative relationship, but many times they don't. And so by calling it, for instance, just the microbiome and that it's an extended phenotype of the host, it gets really dangerous because they're under very different selection pressures. So I think that's something that we want to maybe spend more time thinking about. So microbes as masters of manipulation, that's what I want to talk about now. And specifically, I want to talk about fungi, right? And I get fun of the way, now I can't remember. I say fungi, but everybody else says fungi. I always forget which one I say and which one everybody else says. I think I say fungi, but now I've gotten so sensitive about it, I can't remember what I believe in my heart. But I want to spend the rest of the time talking about that stuff and a particular type of fungi that's called arbuscular mycorrhizal fungi. And I mean, if you haven't heard of I'm getting goosebumps, and I've been studying it for 18 years. It's embarrassing, but it's incredible because it's everywhere, right? It's everywhere. So 80% of all plant species have this fungi growing in their root systems. Yeah. So if you were to go out and pick up a plant, for sure. Maybe like your Arabidopsis, some of our crops don't have it, but most of the plants out there globally associate with this fungus. And it's ancient, right? So I was talking about the legume Rhizobia symbiosis, 60 million years old. This guy, 450 million years old, right? So this fungus was actually key in allowing plants to colonize terrestrial land, right? So these guys, older than insects, older than most things. What you need to know about the fungus is that they're what's called obligate biotrophs. That means they cannot grow without a host plant. They get all of their carbon, every bit of their carbon, from the host plant. So they are completely dependent on colonizing a root system and getting the sugars from the host. What they do is they form these really complex networks underground, and they connect multiple different plants above ground. There's a lot of work showing that they're conduits for communication, but basically what we study is the trade between the plant and the fungus. And what they're doing is they're trading. They're actually out there scavenging mostly for phosphorus. They're looking for phosphorus in the soil, and they're trading it for sugars from the host plant. Now here's one example of how they manipulate the host plant. So for instance, if you've got a plant there and it becomes colonized by mycorrhizal fungi, they actually downregulate the genes of the host plant. They downregulate their direct uptake mechanisms. So imagine you're a host plant, and you can get phosphorus yourself. You're in the soil, you can get it up. The fungi come in and basically manipulate the genes of the plant to say, don't worry about it. I'll give you the phosphorus, just rely on me. And what they do is they create this very cool forced dependency where the plant no longer can sense its own phosphorus environment, but rather has to rely on the fungi for all of its phosphorus needs. So it's actually able to downregulate the genes of the plant. So this is not, we see this actually quite often in nature, these kinds of forced dependencies. It's fantastic. So this is an example of the ant acacia mutualism, where the ants are protecting the acacia trees from herbivores, and in return they get these very rich food bodies. But there are these acacia trees in Central America, and what they do is that in that food that they provide to the ant, they have this protein that actually alters the ant's digestion so it can't consume other food. It can only consume those food bodies, right? So it creates this forced dependency, this addiction of the ant on those particular food bodies, so it has to defend it, right? All it needs were those proteins to get in there, alter digestion, and then you've got this forced dependency. And so what we see is something very similar in these mycorrhizal fungi, is that it goes in and is actually able to manipulate this market in such a way to advance its own gains. Okay, so now let's take a step back and actually understand how this market functions, right? So if you've got a single fungus underground, it's able to grow for kilometers, right? A single individual genotype can grow for kilometers. And so in that hyphal network, it's connecting multiple different individual plants, right? So it's got connections with many different host plants above ground. Now simultaneously, if you're an individual plant growing in the soil, you're connected by multiple fungal species, right? You're not connected by just one, but you've got a choice. You're colonized by anywhere from like 15 to 20 different genotypes on your root system. So what this does is it actually creates, when you've got this multiple partners on both sides of the interaction, is it creates the conditions for a market to actually emerge. And so what we've been doing for, I guess now the last like 5 or 6 years is, oh God, longer. It's a big task. Trying to understand the rules that govern trade in these networks, right, between plants and their fungi. So I'm going to show a short video of how we think it works, rather than showing data about it. This is a much more interesting way of showing how we think it's operating. Okay, so here you've got a root system of a host. Now you've got the root system, and of course the root is giving the sugars to the fungus, right? Here's the fungus that grows from a spore, and it collects phosphorus and gives it to the host plant. Now you have to remember, multiple genotypes of competing fungi, right, are all trying to colonize this root system. So here's one fungus, makes these long hyphal networks, and creates these very beautiful structures inside the plant where the trade takes place. So here's a potential trade. between phosphorus and sugars. But see over here on this side, there's an effective trade. On this side, you've got a smaller hyphae. And what we find is that when the fungus offers less phosphorus to the host, then it doesn't get sugars in return. So somehow, the host is actually able to discriminate at a very small spatial scale how much phosphorus the fungus is giving. But I think even more interestingly is that the fungus can do it too. So it's connected simultaneously to many different hosts. And who is it going to give its phosphorus to? It's going to give its phosphorus to the host that is providing it with the most carbon. So here's an example of a successful trade. Okay, so but imagine, right, that's just a very simple example of like 3 or 4 interactions, but these are massive networks underground where these interactions are happening, you know, constantly with multiple plants and multiple genotypes of fungus. So we think it looks something like this, where there's different interactions happening across space and time. And trying to understand those dynamics is really hard because, first of all, it's underground. And second of all, it's very hard to trace these nutrients through these very complex networks. So one of the things that we do is we use these systems that we can manipulate very easily. So there are these things called root organ cultures, right? And root organ cultures are fantastic because they're very manipulatable. Basically, it's a root system that has no top, has no photosynthetic top, right? So it's just growing in a carbon that only the root can use. And that root gets colonized by these fungi. And so what we can do is we can create systems that we think sort of mimic underground in a system, in a Petri plate, so we can actually visually follow what's happening. So what we see is that, for instance, we can make a host more cooperative by growing it in a higher carbon environment. We can take a fungus and grow it across different Petri plates and add high levels or low levels of phosphorus and see how it trades those nutrients. And what we find is that consistently both partners, not just the host, but the fungus too, can actually detect the different partners, discriminate among a high quality and a low quality partner, and reward the better partner. And so what we think is that you know, a market mechanism, it can help us explain how these behaviors emerge. Now, I don't want to, by any means, say that this is new, right? I think sort of, you know, human behavioral science, evolution sciences has led the charge in trying to understand biological markets in nature, right? And we've known that these kinds of markets exist, for instance, in primates. But I think what's interesting is that there's been this assumption that you need sort of complex reasoning to be able to bargain. And now what we're trying to do is sort of strip that away and say, okay, if we know that there's no sort of complex reasoning, what is economics at its most purest form when you've just got trade and environment? And what we see is that the microbes are actually, they're able to evaluate the potentials of their partners. They can sense the context of decisions. I mean, right now we're trying to understand, are decisions absolute, for instance? Is it just a certain amount of phosphorus for carbon? Or are they relative, depending on what's going on across the whole network? And how is that information transmitted across an entire network? Those are the kinds of questions that we're asking. And it's, you know, it's not just, this is a paper that came out a while ago now, but it's not just these fungi that we think are doing sort of these kind, that we can study in an economic way. There's lots of different microbial systems where we see this kind of trade in the sense that the microbes are able to sense context and make strategic decisions based on that context. Now, there are problems. There are definitely problems that are emerging using an economic framework to study microbes. A lot of biologists don't like it. And one of the biggest, I think, misconceptions, where we published a paper recently about this because there have been so many misconceptions emerging from using an economic framework to study microbial trade. I think one of the most important ones is that it's not an analogy, right? So I think a lot of biologists are saying, okay, well, you're just doing this, but you're just trying to draw an analogy. And actually, that's not true at all. What we're trying to do is use a predictive framework that economics can give us to study trade strategies. And I think the second point is particularly important because what this does is it actually gives us testable predictions, which we didn't have so much in ecology and evolution before. It gives us actually testable predictions about what an organism should do in space and time when it comes to a particular environment. So we can actually test the predictions about these exchange patterns and how they vary across space and time. So it's not just an analogy, we're actually using the predictive framework. The second problem is it's been co-opted by many people to say that there's this beautiful sort of socialistic network underground where organisms share resources. And there's a lot of, even books coming out about, the secret life of plants underground and things like that. And showing, for instance, suggesting that because plants are connected by this network, that they use it to share nutrients among the sick and the struggling and warn each other of impending dangers. And I can talk to you about the difference between a signal and a cue, right? I mean, an organism, might pick up a cue through the network that another plant has been attacked, but that doesn't mean that plant is actually signaling to that plant saying, quick, quick, run, there's a bad thing coming, right? There's a big difference. You can definitely pick up information from a network, but that doesn't mean that an organism is trying to warn another organism. So that's the other second problem. And we see this actually in one of the experiments that we did recently, where we looked at how plants that were not the same genotype, what would you expect for their contributions to a common fungal network, right? If they're different genotypes, what would you think that investment would be? I mean, from an evolutionary standpoint, we thought that there'd be strong selection pressures against hosts who actually invest in a common network that could help a competitor. But we can test that now because we can look at these networks and see how much hosts invest. And this is just a simple graph where you've got the monoculture prediction, so what we would expect if the hosts were in two of the same species. And what we would find on the y-axis is the abundance of the hyphal network, right? So basically you can see that there is a decrease in the amount that the hosts invest when they are not paired with a relative. So again, what this tells us is that it helps to look at it from a social point of view, that the size of the fungal network is going to depend who else is in that network. Okay, we're going to skip over that. Okay, and the last bit, the last 10 minutes, we're at 35 now, I think, is maybe it's less interesting to you guys, but I'm really excited about it, so I'm going to show it to you anyway. And this is this idea of actually being able to visualize trade, right? So, the kinds of experiments that I've been describing, you can think of it as like, there's an amazing poker game going on, and then, I'm the scientist, and at the end, I just get to see everybody's cards, but I didn't get to see anything that happened in the middle, right? It's just this outcome, and it's really unsatisfactory because you put in these different conditions, and then you wait, and you just see who won. It's very simple. It's like big plant, little plant science, right? It's not, I want the dynamic stuff. So we've tried to develop this technique where we use what are called quantum dots that we can attach. These are nanoparticles that actually fluoresce in different colors when you expose them to a UV light. And we attach them to nutrients. So we can color nutrients different colors. Like this is, to me, this is really exciting even as I'm saying it. And we've been working on it for so long. But what you can do is that then it allows you, in theory and almost in practice, is to actually, so you can tag nutrients and then visually monitor them through the network. So it's just like the video, even though we made the video first and now we have the technique. It's meant to be. But we can do this in real time, right? And then you can look at different parts of the network and see sort of the trade dynamics. So imagine even two colors of phosphorus. We can have really rich areas of phosphorus, and color that blue, and then we can have really poor areas and color that red, for instance. And then we can see how those networks are, how those resources are traded and shared across the network. So for us, this seems to have some exciting, that's the chemistry. But the plants, they light up, right? This is amazing. I mean, not quite like this. There's a little bit of photo editing here. But like, if you expose the plant to this nanopart, these quantum dots, then you can actually see the leaves fluorescing. So what that means is that you can, all of this sort of hidden trade, you can start to see in the actual plant itself. And this is what it looks like in the hyphae. That's amazing. Again, so the bigger question is sort of trying to understand the rules that govern trade in these networks. And what I want to know is, like, how do they sense their immediate environment, the fungi, and then how do they integrate that information to inform their trade? Because they're trading with many different plants at many different points in time and in space. So we really want to try to understand how that works. So one of the things that we're trying to do now, we're really interested in this idea of inequality, right? So how does a fungus deal with inequality? How does it deal with being exposed to really high resource patches and really low resource patches? So this is, for example, some of the experiments that we're doing right now. You've got a host up in the upper compartment. That's just the root. You can't see the fungus. But what it does is it grows into two different compartments, and those are physically separated. And it can't grow through the middle, what would you call that, like the 6 o'clock line down. And so then we vary nutrients across that network, and we see how it's traded. So, you know, obviously the control would be to put nutrients in a 50-50 pattern. The next would be 30-70. The next would be 90-10. Now, we found something that I think was very unexpected. What we had assumed was that the fungus in the high resources would trade more with the plant, get more in return, and grow bigger. But instead, we found that it was transporting the nutrients across the entire network. So we started to think about, okay, well, trade and how it works. And you know, in economic markets, it's true, you can either sell your resources locally, or you could potentially move them somewhere else where they may potentially have a higher payoff, a higher gain. Well, in fungal networks, it works very similarly, right? You can get a deal, you know, you can trade right where the nutrients are in this high resource area, or you can move it across the network. But imagine if you're in the high resources, do you think you're going to get a very good price in those high resource? No, right? So what we saw was the fungus is actually picking up the nutrients in the high resources and then moving it to a poor patch. Right? So it's picking it up, and it looks like it's moving it to this poor patch. So this is a little bit of a confusing graph. But it's, you know, you've got your phosphorus in its origin compartment, right? So the pink is in the pink, and the blue is in the blue. And then what we're looking at is how much of the pink moves across the network. And it has to be, there's no diffusion, right? Everything has to be up and over and through the fungal network. And what we see is a significantly more net movement to the poor side. So there is some movement from high to low, but there's also a net movement to the poor side. So we're trying to understand these dynamics. Just to give you an idea of sort of this is what it looks like in real time, right? In real time, they're moving these nutrients in such a cool way. And so what you have to see is how often it changes direction, right? So you're looking at a lot of different things. One, you're looking at what we call bipartite movement. So carbon is moving one way, phosphorus is moving another, but then the whole general direction of the cytoplasmic stream is also changing direction. Right? So it seems like it's coming down to the left, but then all of a sudden it will reverse and go back to the other side. So that's just a visual of sort of what we're seeing. Here's another picture of the network, and so what we're doing now is trying to measure those velocities. We're working with the Physics Institute here. We're trying to look inside the hyphae, measure how fast they're moving nutrients and how often they switch directions. And it's actually quite flexible. It seems like our first numbers are that the flows within the individual hyphae, they're reversing directions every 5 minutes or so. So they'll be going in a stream and then they'll reverse direction. So what are they doing, and how do we understand it? that's the next chapter. We'll see what happens. But the hypothesis that we're working with right now is that the fungus can actually capitalize on this value difference and actually move the resources to where it can get a better deal. Okay, I think that's it. Yeah. So the last slide. I mean, you know, it's great to talk here, and I'm hoping for some easy but good questions. But I do think that there are sort of general laws in nature, right, about how cooperation emerges. And I think that it's important to remember that, you know, it looks harmonious, but I think under all that cooperation is conflict. So it's not the cooperation that we need to learn from. I think it's actually the conflict and how organisms in nature actually deal with that tension. And so we saw that, you know, compartmentalization is incredibly important in stabilizing cooperation. This irreversible dependency, you know, for good or worse, right? Organisms becoming one so that there's really low conflict. And this flexibility in trade where organisms can actually respond to local resource use and move things accordingly. Here's all of the work was done by an amazing group of people, both at Oxford and at Amsterdam. And these are my funding bodies. Thank you so much.
Well, first of all, thank you so much. This was really fantastic. I mean, it's like the best that we hope in terms of crossing disciplines.
Thank you.
No, wonderful, wonderful stuff. So many questions, but in short, can you tell us a bit more about the irreversible dependencies?
Yeah.
The context, the trade-offs, when does it make sense, whether you think it goes along with increased complexity, because like in the case of mitochondria, you might imagine that it's a feature of increased complexity?
Yeah, I don't know how it relates to complexity. I mean, we do see a few examples of mitochondria defecting, right? Where there's these, you know, rogue mitochondria that actually go bad, and that can lead to enormous problems in the phenotype of organisms. But yeah, I mean, what you just asked is sort of like a PhD. We want to know too, right? What is this about irreversible dependency? I mean, I think what we're trying to do is just identify the cases that are happening now by there's just such a wealth of information about individual host symbiont relationships, but trying to collate that across the tree of life is a bigger project. But if we can do that, there's really cool phylogenetic methods now. I mean, it's amazing what you can do now with phylogenetics and statistics. So it's still just modeling, right? We don't really know what was going on back then, but the statistics make it look like it's getting, you know, that we've got a really good resolution for what's happening back there. But the idea is just to collect it and answer those exact questions, right? What is it? Is it the host biology? Is there something about the host that sort of drives this irreversible dependence? Does it have to do with the environment? I mean, people, for instance, with rhizobia, this is a great example. The hosts do really depend on rhizobia for nitrogen. It hasn't become an organelle. Right? And people are trying to engineer rhizobia into things like corn, and it doesn't work, you know. So, and that could be vertically transmitted through the seeds, but it's not. So, you know, there's all of these sort of big questions. But if we can find some of the patterns, I think looking at genome size is very important. So that's sort of what we're doing now is trying to figure out, okay, if we see a small genome, is that the result, is that the consequence, or is that the driver? Like if you already have a small genome size, do you tend to get into a more dependent relationship or not? Yeah.
Thanks so much for a really great talk.
Thanks.
So my question is a little bit more about sort of the sociology of science around these issues. So I've noticed that there seem to be sort of two general groups who are approaching questions about sort of microbiome host interactions. One is sort of a focus that's very much on sort of the evolution of virulence, maybe looking at how microbes might evolve to be less virulent, but not necessarily how they evolve to be beneficial. And then there's a group that is sort of looking at it from the perspective of this sort of extended phenotype, the holobiont idea that you know, individual and all the microbes are a unit of selection, which we know can't really be true based on first principles. But it seems like these two communities aren't necessarily interacting and coming together around sort of creating models or experimental systems to address these questions. And I'm just wondering if you have any insight into why these two approaches are, simultaneously existing and not interacting.
Yeah, no, I also am curious about it because, it seems to me that they both have very different goals because, the more that we study the microbiome as a harmonious part of our organism, which a lot of it is, a lot of it isn't, but a lot of it is, then we can really use it for biomedical research where, with the virulent stuff, it's also quite different, right? They're just coming from a very, very different perspective. And I think in the virulence research, actually, they are studying it under, very nice, strict sort of evolutionary framework. I think what we need to do is have the microbiome community, go to study more evolution, potentially. That's a big statement, I'm not sure. There's a lot of really good people doing microbiome research, that's evolutionarily informed. But there could be more, right? Because then I think it's that tension where we can manipulate it, right? We see this in agriculture all the time where, you get a really good response from a particular community of microbes and, and then you go and put it on the plant and it doesn't work, right? But that's sort of evolutionary about is like, what is happening with the competitive interactions between the microbes themselves. But we're getting there, but we're just, we're not there yet.
So really, really cool work. Sort of two, maybe three related questions.
Wow.
One is
take notes.
with intergenomic conflict, looking at different transmission systems. If you overlay this on the system, how far have you gotten? What do you see? A second sort of related question is when we were doing intergenomic conflict work. I remember that this was long ago, right? The fungi were extremely bizarre in that there were lots of systems where you had genomes without cell walls. So multiple different genomes.
Yes.
And
I can talk about that. I'm not sure I can talk about the first one, but okay, so number two,
And then the third one, which is possibly related, is logically in a market system, it would be possible for different fungi are not just competitive, but you might think that one that was better networked to a sugar source might trade the sugar to another fungal system.
Yes, I can answer that one too. Okay, so I'm not going to skip the first one because I don't know. The second one, I didn't even get into the genetics of the fungi. It's incredible. So if you've got an individual fungi, just like you said, they're aseptate, right? Which means that they don't have cell walls within the fungi. So it's an open pipe system. But they've got multiple genomes across that individual. And there's a huge debate about the level of diversity within an individual and how stable it is. And is it real diversity, or are we just seeing different SNPs or something like that? But it's exactly like you said, there's this conflict within an individual. So we're trying to basically understand, are there competing genomes, and how does the plant, is it, for instance, at the actual spot where they trade is called an arbuscule, if there's many copies of the genomes in that particular arbuscule, is that help populate the whole network with the more cooperative genotype? So there's a lot of really cool theory going on, but it's a lot harder to study empirically. And the second one they would be great if fungi there's so much to say. But there's plants that are parasitic plants that do not photosynthesize at all, right? So they can't give carbon, and the only way they get carbon is parasitizing it off the fungi. So these plants are acalorous, meaning they have no chlorophyll, so they're just growing. They're usually pretty small. But what they're doing is they're stealing carbon from the network, which is actually produced by another plant. So it looks like they're stealing from the fungi, but the fungi didn't produce the carbon, so it's actually... So we know that there's ways that there's these multi-trophic ways that the carbon can be moved through the network that is not fair, so to speak. We see in some of this work with the poor and rich stuff, we see that the fungi growing in very even environments tend to store the phosphorus more. Right? So one of the strategies we think that they're doing is that they're growing out to the soil, they're picking up as much as they can, they're depleting the soil, but then they're storing it until they get a better price, right? Which is, so they're holding on to it and kind of speculating. But again, showing that biologically, right now it's just a story, but we definitely see them storing the carbon under different resource conditions, right? And it's not just the absolute level of resources, it's how the resources are spread across the network, right? So if you can keep the absolute level of resource the exact same, they still show a different strategy if they're in different spatial patterns.
Can you handle one more question?
Yeah.
I was intrigued by your inequality experiments, and I was wondering if you could say a little bit more about the transport of nutrients. I was thinking that some sort of null model whereby they simply float in Brownian or meaningless motion would still net go from rich to poor. So whether it's useful to say they're doing more or less than some null model and what that null model should be. And then relatedly, are there costs, measurable costs of transporting this stuff that increase with distance?
Okay, for the first question, I can answer, and thank God for physicists, right? Because they've done all the calculations for things like passive diffusion because of Brownian motion, right? So it's moving much, much faster and switching directions much more rapidly than under null models of diffusion. So great question. There's like 7 pages of supplementary materials to, to explain that I don't understand. But the question is, we did that. The second question is about, what is it again?
Costs of transport.
Right. Okay. So we know that it's active transport. We know that it's not just diffusion, and it involves chitosins. And so there's some kind of movement of the cytoskeleton. So it has to use ATP. But measuring those costs, like if you have any ideas to how to try to measure those costs, we've got a few ideas, but we want to get at that because, of course, right? I mean, if you're moving resources to an area where you can get a better price, it's not a better price if it takes lots of energy to get there. So we have to measure the cost, but we're finding that difficult. Yes? Great.